Okay, and Dr. Kayode says that he's lecturer at Federal University of Agriculture, Zuru, Kebbi State, Nigeria, at the Department of Animal Science. That's pretty interesting. And we also have Komal introducing her to us, and she's pursuing MSc in biochemistry. So we do have a couple of MSc biochemistry students with us, and a lot of plant biotechnology enthusiasts. I'm also curious to know what is your current level of knowledge of transcriptomics? Is it beginner? Is it moderate? Is it advanced? All right, so with that, let us begin with today's webinar. Hello, everyone. A very warm greeting to one and all. My name is Sri Gauri Krishnakumar, and I would like to welcome you all for today's webinar on omics logic, transcriptomics data analysis for biomedical data. This is an online mentor-guided program on next-generation sequencing data analysis in application to gene expression data with project examples from infectious diseases, cancer, and neuroscience. And this program will introduce students to real-world applications of RNA-seq data analysis in biomedical research and provide hands-on skills and logical background to extract insights from gene expression data. And in today's webinar, we will first learn to sign up on the Omics Logic Learn portal and learn how to navigate the various courses, associated resources, objectives, and outcomes. Finally, the participants will be introduced to the various subscription options for the enrolling in the program. So before we begin with today's webinar, let me also take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. And as a part of the program, students will have access to the Omics Logic training that has been completed by over 24,000 participants from 187 countries in over 300 workshops. And due to this fast growth, our team is working with local and regional coordinators that are helping refine our local program logistics and adapting them to the needs of students and researchers around the world. And this training has been completed by participants in 6 different specialization tracks that includes oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. Students will also have access to the Omics Logic resources. But how does it help advance bioinformatics teaching? Omics Logic is a portal for practical and theoretical learning of bioinformatics. It takes a combination of training modules, data analysis tools, curated project data sets, and interactive sessions with mentors to give the student a clear path up the Bloom's taxonomy pyramid. And no matter what the user's initial experience with bioinformatics is, Omics Logic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large datasets, and introduction to basic coding languages like R and Python, or even beginning a data analysis project. This also comes with an access to the omics logic courses, which includes courses on bioinformatics, precision medicine, multi-omics that includes genomics, transcriptomics, metagenomics, epigenomics, data science that includes using R and Python, as well as cheminformatics. And example projects of different fields which you could learn from and replicate for your research work. The users will be given directions to access the tBioInfo platform for bioinformatic processing and analysis of data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run the bioinformatic pipelines, and different stages of analysis are performed in different sections on this multi-omics platform. Now, to view the various courses, example projects, and student projects, you need to first sign up on the Omics Logic Learn portal. Before that, let me walk you through a brief demo on what a completed profile on the portal looks like. So once you sign up on the portal, these are the features that you need to update on your account. That is, a completed profile will have a profile image, full name with appropriate capitalization, link to your social media handles. This could be your Facebook, LinkedIn, Instagram, or ID, or even Twitter, and a brief bio about yourself that will consist of your educational background and research interest. The mentors can view your progress under the Activity tab. The courses you are currently completing will appear under the Courses tab. Finally, after the completion of your course, the certificates will appear under the Certificates tab. So with that, let us begin by signing up on the Omics Logic Learn portal before proceeding with further details about the program. So for that, let me share the link in the chat box for signing up. All right. So here's the link for signing up on the portal. So for those participants who are new to the Omics Logic Learn portal and do not have a previous account, you can create an account on the portal by simply entering your email ID, your password, and simply click on, first you need to click on create an account option that you see here. And then here you will be asked to enter your name, email ID, and password. And you can also have the option to sign up using any of your social media handles. This could be Google, Facebook, Apple account, GitHub account, or even Twitter. And with that, I'll pause for a couple of seconds so that the participants can sign up on the portal. When you sign up on the portal, please put a yes in the chat box. And for those participants who already have an account on the portal, you can simply click on Login Now option and simply click on Sign In. So those participants can put a yes in the chat box so that I know they already have created an account on the portal. And I'll help you navigate through the various courses and the program details that are there. So I'll pause for a couple of seconds. Okay, Akash says done. Sarah says yes. I'm waiting for other participants. We have Pami, yes. Taliemi says done. If you're facing any technical issues while signing up, you can mention that as well, and I'll be happy to guide you. All right, Tolano says yes. Andrea says done. I'm expecting more yeses and dones from the participants, and I'll show you the various courses and example projects that are there. All right, so meanwhile, the rest of the participants are signing up. I'm going to sign in since I already have an account on the portal. All right, so once you sign in, you'll be able to see the various courses and example projects that are there on the portal. So if you're looking for a specific course, you can simply enter the topic that you're interested in. So for example, if you're interested in transcriptomic, simply enter the keyword transcriptomics, and that will show you the various courses and example projects that makes use of the concept transcriptomics. And let's say you're interested in machine learning. For that, you can simply enter the keyword machine learning, and that will show you the various courses and example projects as well. So now coming to the part where I was showing you how to update your profile on the portal. For that, click on the tab that says welcome back with your email ID. So once you click on this, you will see an option that says profile. So when you click on this icon, it will show you how you can edit the various details on the portal. So for instance, you should have a profile image. Your name should be in proper capitalization. You should link your social media handles. For instance, I've linked my LinkedIn account and my org ID, and a brief bio about yourself. So in my case, I have mentioned my educational background and my research interests. And under the activity tab, as I previously mentioned, the community manager and the program mentor will be taking a look at your activity, coursework activity, and the various courses that you start picking up will be displayed under the courses tab. And once you upload a research project on the portal, that will be displayed under the projects tab. And once you enroll in any of the programs, for example, the Transcriptomics program, that will be displayed under the programs tab. So I'll pause for a couple of seconds. Does the participants have any queries? Otherwise, I'll proceed next. All right. So now I want you all to click on the Programs tab that you see here. And once you click on the Programs tab, that will take you to the various program lists that are there. So here, when you enter Transcriptomics, you'll be able to see the main transcriptomic data analysis program page. When you click on that, you will be provided with various details for the program. So I'll share the link in the chat box so that the participants can view it as well. Now let us take a look at what are the various details that are listed on the program page. So first of all, you can see that the program commences on 20th December and ends on 30th April 2023. And as you scroll below, you'll be given a program overview of the transcriptomic data analysis program. So you can briefly go through and understand what are the various topics covered in a summary wise. And when you scroll down below, you will be mainly seeing 4 key highlights. That is, one is that you will see the session dates for the program and the calendar that is displayed here. You will also see the various session titles. So here, if you scroll down, you'll see all the various sessions that are going to be conducted as a part of the program. In addition to this, you will also see the various topics that will be covered in each of these sessions as well. And apart from that, you'll also see the associated online resources that the program mentor will be making use of as a part of the program. So all of these are hyperlinked. You can simply visit the link and that will take you to the lesson or the coursework that will be covered during the session. So you can browse through and go through the program page at your own pace. So with that, let me go back to my presentation and let me walk you through the various associated resources that you will be getting as a part of the program. Does the participants have any queries so far? So you can let me know and I'll be happy to guide you. Has everyone visited the link? Has everyone created an account on the portal? Okay, all good. All right then. So as mentioned earlier, one of the associated resources that you get as a part of the program is access to the Omics Logic Learn portal, where you saw the various coursework and the example projects, as well as student projects that you'll be getting a chance to take a look at. And apart from that, you will also get access to the T-Bioinfo platform. So we know that high throughput transcriptomic data analysis increasingly rely on bioinformatic pipelines, but biologists often lack the required technical expertise and often struggle to work with transcriptomics pipeline. That is why we offer easy to follow transcriptomics pipelines that are color coded, as you can see in the animation, that will help you derive meaningful insights from complex data sets. So this is the interface of the T-Bioinfo platform. I'd like to request my colleague to share the link in the chat box to the T-Bioinfo platform. So you can take a look at the interface and what are the various sections that are covered as a part of the TBioInfo platform. Now, let us take a look at what else is there. So again, data visualization options often empower biologists and researchers to visually explore and analyze transcriptomic datasets, such as tables of gene expression in a short and intuitive manner. That is why we have also incorporated interactive data visualizations in our coursework, such as the multi-QC report, bar plots, volcano plots, and heat maps, so that it is easy for visualization. And another important associate resource that you get as part of the access to the TBioInfo platform is the interactive dashboards. So to bring data and biology closer together, we have incorporated interactive dashboards for visual exploration of complex data using the open source Plotly. And most of these dashboards are already available to use by our researchers and are embedded on the Omics Logic Learn portal for the students while completing their coursework. And another important access that you get is the Omics Logic Code Playground, which will help you develop coding skills while learning popular packages in R and Python to visualize, annotate, and analyze complex patterns in the gene expression data. And with so much data available on any biological topic and question, completing a program seems like a great opportunity, but it can be challenging as well. That is why you also have the opportunity to seek mentor guidance throughout your program to help you along your learning journey. So now when you enter the keyword transcriptomics, there will be 3 major courses that will pop up. That will be Transcriptomics 1, which makes use of the TBioInfo platform, and you also will see the Transcriptomics 2, which makes use of the R programming language, and you will also see Transcriptomics 3, that makes use of Python coding language. So for those participants who do not have a prior background in coding and who's completely new to the field of transcriptomics, then Transcriptomics 1 will be the best course to start with. So in this webinar, I'll be covering more about the highlights of Transcriptomics 1. So what are the various objectives of this course? That is, when you enroll for the program and when you get access to the Transcriptomics 1 course, first of all, you will learn about the key terminologies and basic concepts in transcriptomics. And this will be taught through Lesson 1, Biology of Transcription, and Lesson 2, RNA quantification. Another important objective of this course is to introduce the participants to next-generation sequencing with specific focus on RNA sequencing, and that will be covered as a part of Lesson 3, RNA sequencing and next-generation sequencing. So far, we covered mostly the theoretical concepts. Now, the course will take you one step further and help you understand the logical steps and learn how to run bulk RNA-seq pipeline. And this is where you will first of all start using the TBioInfo platform. For this, you will be introduced to the lessons 4, analysis of raw RNA-seq data, that makes use of logical steps, and lesson 5, practical RNA sequencing. And then you will also learn how to carry out the downstream analysis using simple statistical comparison. And here you will also learn about PCA and quantile normalization. So here the associated lessons that you will be covering will be data preparation for downstream analysis, lesson 7, practical normalization and PCA of gene expression data, and finally lesson 8, statistical test. And another important objective is to introduce the participants to differential gene expression analysis and perform differential gene expression using the DSEQ2 option and regression analysis using the TBioInfo platform. So for this, you will be making use of lesson 9 and lesson 10. Lesson 9 will introduce the participants to differential gene expression and gene enrichment analysis, and lesson 10 will introduce the participants to regression and factor regression analysis. And finally, you will also have the opportunity to conduct advanced statistical methods using unsupervised and supervised machine learning techniques, and you will learn to derive and interpret important biological insights after the analysis. So for this, you will be taught through lesson 11, cell line data and preparation, lesson 12, unsupervised machine learning and clustering, and lesson 13, supervised machine learning, lesson 14, which we will be covering in depth about the feature selection. And finally, we have lesson 15, which will teach you about how you can go one step further to apply your knowledge that you have gained so far to example projects and even carry out an independent research project on a topic that you're interested to work on. So now you might be wondering, after all the coursework completion, what comes next? So for all of that and more, I'd like to pass on the stage to Dr. Raghavendran Lakshmi Narayan, who will be your program mentor as well. So the stage is all yours, sir.
Thank you very much, Srigauri. Let me set up my screen sharing. And I take this opportunity to welcome all of you to this webinar. And yeah. Okay. Can you see my screen? And am I audible?
Yes, sir.
Okay, thank you very much. So welcome to today's webinar. So as Srigauri has briefed you about the resources that you're going to be going through throughout the session. So my part of today's webinar is going to focus on what are the data sets that we are going to deal with, and what are the questions that we are going to ask with the data sets, and some information about sorry about that. Yeah, some information about what are we going to discuss using example projects that we have prepared for you to go through the process of using a transcriptomics data analysis to answer key questions related to disease biology. So that's the main focus with this specific section. So let's, without further ado, let's move forward. Yeah, great. So what have we learned so far? So we know that the development and the advent of modern genomics has provided completely new ways to link targets to diseases and to validate these targets, right? And the result is, for example, a potential for a new era of investigating the disease progression through regulation of gene expression. And these developments actually provides a mean to mimic the mechanism by which our bodies can exert regulatory control over a range of normal functions and also functions in disease or functions in altered cell state. So in this program, we will discuss, we will review, and we will demonstrate using data sets that are associated with popular publications and public repositories to demonstrate you different approaches that can be employed for using transcriptomics data analysis to understand various aspects of chronic disease biology. So how are we going to do that? Let's proceed. During this journey, we will also learn about basic and translational research that is specific to oncology, right? Where the patient-derived cell lines, which is a model system, and patient-derived xenograft models, which is a model system inside a mouse models, right, offer extremely good and rich collection of data to understand the treatment of cancer in patients, to understand diagnosis of cancer generally, and also to specialize in precision medicine or personalized medicine. So we will be using data sets from patient-derived cell lines, and we will be using data sets from patient-derived xenograft models, and we will be using data sets from patients themselves. So all these three different data sets will give you an overview of what are the methods that you have to understand and that you have to compile to analyze the transcriptomics data from three different sources to all of them culminating into one single aim to understand disease biology of tumor cells, right? Let's move forward, and in one of the examples, we will take a data set from publication modeling precision treatment. Excuse me, I think yes. So we will take a data set from a well-known publication that is modeling precision treatment of breast cancer, right, which was featured in Genome Biology in 2013. This is a very interesting publication because the authors actually assembled 50 different breast cancer cell lines and reported a whole and represented a whole spectrum of breast cancer variations, including, for example, results associated with treatment responses, results associated with various molecular pathology and disease progression, etc. So how did they do that? They integrated data sets from transcriptomics data. They integrated data from genomics analysis, genomics data, and also proteomics data, along with treatment response data, to multitude of different machine learning models to understand generally the transcriptional profile and to understand omics data profile that is going to explain commonly used explain responses of this tumor to commonly used drugs. So this data set and this publication is very, very rich, as I mentioned, because it uses 90 different drug response signatures, and it uses, for example, 7 different molecular profilings, and it also uses 84 lines of cell panels, right? So in our analysis, or during the course of this program, in the sessions, we will be using data from this study and address some of the key questions using commonly used advanced statistical techniques like unsupervised and supervised machine learning analysis. So in the unsupervised machine learning analysis, we will use transcriptomics data to understand categories of breast cancer that can be obtained or that can be identified using normal clustering techniques like hierarchical clustering, like k-means clustering. And you will not stop there. You will further proceed into comparing the results from different clustering methods and how they interpret transcriptomics data to give you an overview of the categories that is present for breast cancer. And we don't stop with that. We also use multi-omics integration, like we integrate transcriptomics data along with the treatment response data in the supervised machine learning analysis to predict treatment response and to understand how transcriptomics data can be used to investigate tumors' response to some treatments. So that we will do, right? And overall, this project will actually help us learn about how to analyze various omics data types, how to integrate them, and how to associate them with the phenotype, that is a response to the treatment, and also different categories of breast cancer, right? So this gives us a perspective on drug screening. This gives us perspective on precision medicine to precisely diagnose the stages and precisely diagnose the breast cancer categories of the patients. So following which, we will make use of another publicly available resource that everybody knows about, the Cancer Genome Atlas, where we can actually, we will show you how to download and how to access data from this public repository, and how to, for example, compile the data so that we can perform some transcriptomics data analysis that is generated or that is collected or compiled from patients' data, right? We will show you, we have a separate session that focuses on, or part of the session that focuses on how to access data from public repositories and how to pre-process them and prepare them for further analysis. So once some of the pre-processing is done, basically what are we trying to do is we will take samples from, let's say, liver cancer, like liver hepatocellular carcinoma, and then using the TCGA data from this cancer, we will try to link, or we will try to relate the patterns observed in the transcriptomics data of the liver cancer patients to various clinical data available on those patients, like hepatitis B infection complication, like the age at which the initial pathological diagnosis was done, like BMI, obesity, or like also geographical locations based on the severity or based on the complications of the hepatitis B infection was actually found. So these different clinical data or different risk factors can be evaluated using, or for example, studying the patterns observed in the transcriptomics data themselves, right? So let's move on. How are we going to do this? So this is not, if you want a comprehensive understanding of different clinical characteristics that actually helps us in generating categories and understanding overall patterns of the hepatocellular carcinoma, then you have to definitely check out the Cancer Genome Atlas paper that focuses on this liver cancer. So in the end, what will we get? We have, for example, scope for integrating several of the omics data analysis, omics data, and to use them to understand, for example, let's say, the transcriptional or transcriptomics landscape of the disease, the transcriptomics signature of the risk factors associated, and etc. So to give you a short demo of what are we going to do and how are we going to focus and what are we going to do with the how to process or what are some of the important findings from these results. We will take a subset of this data, focus on only 20 of the samples, and when we try to understand the patterns available within these 20 samples, we readily see that there is no pattern that one can identify from the principal component analysis as an exploratory data analysis. This is due to a lot of noise that is present due to this data being from the patient resource or patient source, and this patient data has been collected from various different resources, and then we see that generally patient data is associated with increased amount of noise compared to the model system where many of the different controllables can be controlled. But once we use advanced statistical methods like machine learning analysis and supervised machine learning, and from, for example, applying random forest algorithm to this data can actually enable us to use a subset of genes to understand the pattern generally. So instead of using all the genes, which results in a noisy analysis or noisy principal component analysis output, if we use a subset of the genes, we see that the samples very well separate themselves based on the risk factor that one can associate with the data or with the samples. Like we can identify alcoholism and hepatitis B complications, and we can also understand other diseases associated with the patients. So we see, or we have resources, and we have lessons on how to perform this analysis, how to set up this analysis, and furthermore. So we will also use, as a continuation of this same project, how to integrate multi-omics data like gene expression data along with microRNA expression data to understand risk factor again, or to understand alcoholic prevalence in the liver cancer, and that's it. So if you have already access to the TBio to the Learn portal, so you should definitely check this out. And we will also, during the course of this program, spend a lot of time on this project and focus on how to set up the analysis and how to interpret the results, so that you can get an overview of how different advanced statistical methods can be applied to transcriptomics data to understand disease progression, as we already spoke about. So transcriptomics data is not all about using machine learning and with the different algorithms. In fact, most of the transcriptomics data analysis will actually be employed to understand or to elucidate differentially expressed genes in the disease state compared to the healthy controls. So in this project, we will actually explore the transcriptomics data of advanced stage Alzheimer's disease patients, right? Then where we can use the differentially expressed genes, or we can identify the differentially expressed genes and then use that to annotate or use it to associate with the different pathways that we will find that are significantly enriched or significantly suppressed in the Alzheimer's disease patients compared to the healthy controls. We will first, for example, explore the transcriptomics data using normal exploratory data analysis like PCA, box plot, and etc. So subsequently, this will help us in identifying outliers, and also setting up necessary contrasts to perform differential gene expression analysis, from where we can actually scrutinize what are the important pathways that are altered in the advanced Alzheimer's disease patients in comparison to the normal samples, using overrepresentation analysis, gene enrichment analysis, and gene set enrichment analysis can also be performed. So some of the projects that we actually discussed, example projects that we actually discussed, are actually available in the Taubot BioInfo platform, as briefed by Sri Gauri already. So in the platform, you can actually go to these drop-down box, that is our drop-down option next to each of these analysis option. That is going to open you up to a lot of demo pipelines. And in these demo pipelines, we can actually focus on, in these demo pipelines, we can actually focus on how this analysis is set up. And then once you get a practice of how to set up this analysis, this actually gives you, thank you, Shrivam. This actually gives you access to all the output that you can download, and then go through the interpretation that is shared or that is discussed along with the courses associated resources. Let me stop and reshare and show you where to access these demo pipelines from, so that it is easy for you to gain an understanding of how to access this. Let me stop share here and then resume share when I set up this analysis. Okay, can you see my screen?
No, sir, not yet.
Yeah, wait a second. I think it's still working on finding my screen. I think this time it should be successful. It actually found it. Can you see my screen? I have opened up the server, TBioInfo server.
Yes, it's visible.
Yes, thank you very much. So next to each of these analysis technique and analysis methods, we have this drop-down option. This option actually opens you up to several of the demo pipelines, and some of the pipelines will actually use the data sets that we discussed just now. So this will not only give you an insight into how these data sets are used to set up the analysis, and it will also give you access to the output files, which you can download and follow along with the interpretation that is referred in the associated resources. So let's say we have the demo cell lines projects or the PDX project. Let's take this demo cell lines project. If you click on this, it's going to open up the pipeline options to set up transcriptomics data analysis. The only difference here is we have uploaded the necessary input files, and we have set up all the necessary parameters that you will learn how to set up and how to and what to choose during the course of this program. So this is going to give you a practice or a preview how to set up this pipeline. So we start with this. Once I can say yes, then followed by pre-processing methods, which will also be discussed. It's very slow today for some reasons. Yeah. Then it gives you clues, or it gives you highlighted options, which is going to be the theme when you are going to set up a pipeline for your analysis also. So it's going to give you multiple options that you can choose from, and the choice of the option will depend upon the data analysis that you are setting up and the question that you are going to ask. As you can see, after the pre-processing, I have straight away opted to map it onto the transcript. And why mapping onto the transcript? Why not mapping onto the genome? And why not perform isoform construction? These are the options, are the one that we will discuss during the course of this program. And what are the limitations or what are the advantages of setting up one data analysis over the other will also be discussed in detail. So after setting up this, we can end the pipeline. This is a simple pipeline which is going to give you a table that that is going to quantify the mRNA expression levels of the of the samples that we have in the cell lines data set. So you can see there are several outputs that you can download and explore. So to starters, you can explore the multi-QC report as demonstrated by Srigauri, and also the mapping stat, which will give you how good or how well your reads are mapped onto the reference genome. And I'm not going to go over everything, which we will cover in detail. And you have options of expression quantification performed on gene level, at the isoform level, with different normalization. All of this will be discussed during the course of this during the course of this program. So let me again reshare this. If you have any questions, then be prepared with the questions and we will. So with that, I want to pass it on to next stage where, for example, what is it necessary to go through these example projects and understand the steps of setting up a hypothesis, validating the hypothesis, and etc. Right? So those are extremely important for you. For example, let me reshare here. This is not. For you, can you see my screen again now? I think it's gone on to. Okay. It's again trying to find out. Yeah. So as a result of this program, and also as a result of you going through the associated resources, right? Any one of you who wants to start your understanding in bioinformatics data analysis for transcriptomics data can actually start preparing your independent project as we come near the end of this research, as we come near the end of this program. So for your project, you will be selecting the data, you will be selecting the methods, and then which will constitute towards your own research projects, which we have experience of already working with many of the students who have completed the training and have developed interesting research projects that also won award and publications in peer-reviewed journal. So to elaborate on this and also to focus on what is possible and to give you a sample of the research projects that our students actually submitted in our portal and also available as publications and posters in publicly available resources, I invite Srigauri to continue the session. And if you have any questions, I'll be happy to take it, or we will pass the stage back to Srigauri.
Thank you so much, sir. We do have a query in the chat from Faliemi, that is, I cannot access the data in BioInfo platform. So it looks like you are on mute recently. All right, let me share my screen meanwhile.
Faliemi, if you have difficulty in accessing the data in the BioInfo platform, what do you mean by that? If you cannot access the TBioInfo platform, I think you should get in touch with Monica or Spursh.
Hello, Dr. Raghav. Let me answer this question.
Okay.
Yeah, thank you, sir. So, Felimi, if you're not able to access the data in TBioInfo server platform, I guess you are asking that you cannot upload the data, or you cannot run the pipelines or login or registration part. To upload your own data and do the analysis on TBioInfo platform, we have different licenses or different subscription which you have to subscribe for, and then we can provide you with the credentials for the TBioInfo platform to access the platform and do the analysis further and the interpretation part. So I suppose I answered your question. Thank you. Srigauri, you can continue.
Yes, we have one more query from Shruti. As a part of this course, do we have to use the pipeline on the TBioInfo platform, or will we be learning to write code to run locally as well? Dr. Raghav?
Hello.
Yes, sir.
Yeah. So you will be using both, Shruti. We will first start with understanding the logic behind setting up a transcriptomics data analysis using the pipeline option from tBioInfo platform. And of course, analyzing or of course, running a big data analysis for FASTq files, which can easily span multiple GBs and GBs is not possible for everybody. So we have to do the quantification pipeline in the big data bioinfo server, tBioInfo server only. The downstream analysis, you can either continue with the pipelines option in the tBioInfo server, or you can use R or Python's coding options, where, based on your comfort level and based on your expertise.
Thank you so much, sir. So let me proceed with the presentation. So as Dr. Raghav discussed, we'll once you learn the basic concepts and figure out how to run the pipelines, you can apply your knowledge so far on example projects that are based on high impact research publications. And apart from that, you also have the opportunity to carry out independent research projects. So here is a look into some of the research projects that have been carried out by students ranging from high school background to even doctorate students. So the topics range from oncology, infectious diseases, virology, and so on. So here's a look into some of them. So one of the research projects that was carried out by a student was in autoimmune diseases, where she learned to assess the underlying reason for gray matter lesions in multiple sclerosis patients. And in the chat, I'm pasting the link to her project so that the participants can take a look at it. We'll be coming back to this project in the next slide, so keep a note of that. And then another interesting research project was done on oncology, where the participant aimed to analyze novel biochemical interactions before and after administration of drug in cancer patients. And you can have a look into that as well in the chat. I'm pasting the link. And there another interesting research project was done in space omics by a PhD student, where he learned to correlate the effects of space flight conditions on the transcriptome of zebrafish embryos and human astronauts. The participants can have a look into that project as well. Here's the link. And another interesting project was carried out in infectious diseases, where the student focused on TB treatment in human body and how blood transcriptome analysis reveals the evolution and resolution of the immune responses in tuberculosis. Here's the link in the chat. And then we also have student projects in neuroscience, where the student performed a transcriptomic analysis of Alzheimer's disease to understand the difference in elderly population and the younger generation. And here's the link. And finally, we also have interesting research projects on machine learning. Here, the student was trying to examine the transcriptional changes that define different subsets of opioid use disorder patients using differential gene expression analysis and unsupervised clustering. I'm pasting the link in the chat box as well. So as you can see, all of these research projects are in a format that you can share the link and share it with your community or in your resume so that your peers can take a look at it. And apart from carrying out a research project, the students have also gone on to publish their work in in leading journals, such as here is a look into an research project that was done in the autoimmune disease that I was talking about earlier. Here, the student went on to publish a project in immunogenetics and open access research journal, and I'm sharing the link to her publication in the chat as well. And then there's also another student who carried out his research project in malaria vaccine, and that was also published in the Genomics and Informatics journal. So you can have a look at that as well. So now before we wind up, I want to show you where you can find all of these resources, such as the example projects and the student projects. So as mentioned before, when you enter the keyword transcriptomics, that will show you a list of courses and example projects. Another option is to just click on the transcriptomics icon that you see here. And this will show this one will show the various example projects that are there. This will appear under the courses tab um, that gets logged, that is shown when you sign up on the portal. So here it makes use of different concepts apart from transcriptomics, such as machine learning. And here is another interesting example project on the infectious diseases and so on. So now how can you take a look at the various student projects? For that, you can simply click on the tab that says projects here. And here you can see the various student projects that are there. Here also you can simply enter the word transcriptomics or click on the icon transcriptomics, and that will show you the various student projects that are there. So with that, if the participants have any queries, please let me know and we'll be happy to answer. All right, there was one question from Shruti, but it looks like it has been answered. All right, so if the participants are having any further queries, please feel free to share it in the chat. But now I'd like to pass on the stage to Ms. Parshar to share with the students the details for the program registration. So over to you, Sparsh.
Thank you so much, Sri Gauri. Let me share my screen. So could you please confirm, Sri Gauri, if my screen is visible?
Not yet. It is showing started screen sharing. Yes, now it is visible.
Perfect. So hello everyone. My name is Sparsh Adhar and I'm the marketing manager here at Pine Biotech. And I'm responsible for this session to explain you about the resources which you'll have access to once you'll be a part of this program, and then the session topics which we'll be covering throughout the program, and then the pricing of the program and the scholarships. So as you can see that the first session, like the program is commencing on December 28th. The first session of the program will be conducted on December 20th, 2022. And as we scroll down, we can see that there is a program overview. Okay, so here's a program page link in the chat box. You can go through with this link after the session gets over thoroughly. And if you feel any question or you have any further query, I'll be sharing my mail address and also the official contact number here in the chat itself. And you can directly ping me on the mail also, or via call or WhatsApp. So here you can see that this is the program overview of transcriptomic data analysis for biomedical data program. And in this online program, we'll be introducing the participants how they can use the applications of RNA-seq data analysis in biomedical research, and provide the participants with both the theoretical as well as the hands-on skills, so that they get some insights, this extract some insights related to the gene expression data, etc. Now as you move further, you can see here there is this interactive session calendar, and there are some dates which are highlighted on this calendar. So these are the dates when we'll be conducting our particular session on different topics related to this program. And on the right hand side of this calendar, you can see there are different sessions. So the first session, that is on 20th December, it will include introduction to NGS transcriptomic, where the topics we'll be covering are on overview of the omics, next generation sequencing, mRNA biology, methods of RNA quantification, mRNA library preparation, next generation sequencing, Illumina, microarray, Oxford nanopore, and then the biomedical projects using RNA-seq data. Here also you can see that there are some associated online resources which you'll have access to once you'll be the part of the program. And then the second session would be on processing transcriptomics data, where we'll be guiding you and teaching you about the read quality, FASTq, about how you can prepare the expression table, read counts, FPKM, then TPM, gene expression examples would be there, then exon and junction detection, etc. Then again, there are some associated online resources. Then the third is on projects using RNA-seq data. The fourth is on loading RNA-seq data in R and Python, data normalization and preparation for analysis, data visualization in R and Python, exploratory analysis, differential gene expression, then factor regression analysis, biological interpretation, single cell RNA-seq methods, etc. And in the end, we will be guiding. So whatever you have learned throughout the program in your training period, theoretical as well as practically, we will be guiding you how you can transit everything into a real life application by developing a independent research project out of it, or not, if not the project, you can develop a proper research proposal also. So this is what we'll be covering in the program. Now, when you scroll down, you can see there are three different tenures. One is 45 days, another is 60 days, and then is 90 days tenure. So there are some common resources which everyone would have access to if they enroll in the program. And those common resources are, first is the online tutorials. Second is the example research project, which Dr. Raghavendran and Sri Gauri has already explained comprehensively. Then third is the coding sessions. Fourth is the session recordings. Then fifth is online mentor support, online technical support via the Skype group, which will be specifically created for the participants of this program, and obviously the certification after the completion of the program. And then you will have the access to the example cloud pipelines on the TBioInfo server. So these are the common resources which one would have access to once they'll enroll in the program. Now in the 45 days tenure, we'll be focusing on getting you trained uh, in this particular topic and the topics which we'll be covering throughout the program, and through the live sessions or through the online mentor support or live Q&A session, etc. Now the difference between 45, 60 and 90 is, in 60 days you have two different opportunities. The second opportunity, in 60 days, the two different opportunities are, first is one-on-one mentor sessions, and the second is the research proposal. So one-on-one mentor session, for example, if you're going through a particular module of this program and you get stuck somewhere and you cannot wait until the next live session will be conducted, you want to get clarified your queries and question. If the query is small, you can directly ping in the chat also via the online mentor assistance. If not, we can fix a kind of a short meeting of your with the mentor of 20 to 30 minutes, and you can discuss and clarify your questions in that particular meet. Now the second is research proposal. So once your 45 days of tenure is over, the rest of the 15 days you can devote to draft your experimental design. What would be the data set you need to work on, a research question, a title, a reference paper you are following, a methodology, etc. So you need to create and draft a research proposal to present it to your program mentor, and he'll be evaluating your research proposal. If there are any amendments or changes that need to be done so as the research proposal becomes more validated, so he will guide you accordingly. If not, then you can proceed further by developing and working on your independent research project also. And then comes the last tenure, that is the 90 days tenure. So in the 90 days tenure, again, you have two different opportunities. One is obviously the one-on-one mentor session that I explained just now. And second is working on the independent research project. So once your 45 days of training is over, the rest of the 45 days, we will be guiding you, the mentor, the project manager, they'll be guiding you how you can develop your independent research project out of whatever you have learned, whatever you have get trained for, and into a proper full-fledged research project. And the outcome of this tenure would be either a poster presentation in a research symposium or an international conference, or to get it published in a peer-reviewed journal. So if you complete this 90 days tenure with a submission of the project on our portal as well as get it on our portal, you will get a certificate of excellence, as you can see here on the resources. under the tab. So this is how the resources are, and this is the difference between the tenures. Now talking about the pricing part, so right now you can see that the pricing is in INR. How you can change the pricing according to your location? How? So here you can see there is a drop down arrow. Once you'll click on this drop down arrow, you can see that there are different yeah, so there are different currencies which have been which you can see on my screen right now in USD, in Euro, in Brazilian, then in INR, then in Naira, etc. So according to your location, you can change the pricing. For now, I'll be showing the pricing in three different currencies. One is in INR, second is in USD, and third is in Naira, starting with the INR first. So the pricing for 45 days for INR is rupees 7500. Then for 60 days it's 10,000, and for 90 days it's 15,000. And right now we are providing with a 50% scholarship offer, and those who are interested, they can ping us here, on my email address, which I'll be just writing down. Thank you, Shrigori, for the details. So Shrigori has already pasted my details in the chat box. It's marketing@omicslogic.com. So whosoever is interested for the 50% scholarship can either ping me on this particular mail address or can WhatsApp me on the number mentioned, or they can put their email IDs in the chat box itself. We will be texting and mailing you regarding that and providing you with the steps of registration further. Now, showing you the pricing in USD. So, the pricing in USD for 45 days is $150, and then for 60 days it's $300, and then for 90 days it's $450. Again, 50% scholarship is applicable, and then showing the pricing in Naira. So, So the pricing for 45 days in Naira, it's NGN 50,000. Then for 60, it's Naira 99,999, and then for 90, it's 1,50,000. So these are the pricing for three different currencies right now. Those who are located other than these three regions or locations, please change your currencies accordingly, and you can see the pricing and apply the 50% thing, and that will be the amount which is for 45, 60 and 90 days. It's applicable for everyone, and this offer is limited till 25th of the December. So please, if you want to avail this offer or opportunity, please feel free to ping us, and we'll be there to guide and assist you and get started uh, with learning and getting yourself trained in transcriptomic data analysis, both single and bulk. So this was all about the research, the registration part, the resources part, and the scholarship part. So if anyone is having any kind of query or doubt regarding the process, please feel free to ping in the chat box, and I'll be there to assist and guide you. Thank you so much.
Thank you so much, Sparsh. So we'll pause for a couple of seconds so that the participants, if they're having any questions, you can share it in the chat, or please feel free to unmute yourself and ask your queries as well.
Yes, and those who are willing to join the program with a 50% scholarship, and you are interested, so please ping your email address in the chat box. One of our representative will mail you and follow up with you by guiding you how you can register, by providing you with the coupon code for 50% scholarship, etc. Thank you, Alma. I can see you are interested. We have noted down your email address. Anyone else? Thank you, Ashni. Thank you, Shruti. Thank you, Prajesh. Okay, I could see one. So it's Shah Fahid. Okay, thank you. We have noted. Thank you, Akash. So we'll be getting back to you by tomorrow morning, preferably, depending upon which location you are located. I will be mailing you according to the time zone. Thank you, everyone. I could see that you have pasted and you have pasted your. Thank you, Savinder. Anyone else? So I guess Shubham or like any of my team member is noting down all the email address. I guess, yeah, I guess the people who are interested, they have pasted their email address already in the chat box, Sri Gauri. So you can continue. Thank you,
right.
everyone.
Thank you so much. So if you're still left to share your email address with us, you can also share it to marketing@omicslogic.com, and we'll get back to you with a coupon code as well. And if no one has any queries left, we are good to end today's meeting. And with that, okay, we have one query from Shruti. I joined a bit late, so this question might be answered, but as a part of this program, Transcriptomics 2 and 3 also to explore with Python and R, or is it a separate sign up?
So it's included in the associated online resources, Shruti, Transcriptomic 2 and 3. You'll be exploring both. It's not you have to register for a separate program to explore Python and R together with Transcriptomic. It's there in the curriculum. Yeah. You're welcome.
All right, then. So if the participants have no queries, you can leave the meeting. We'll just take a note of all the email ID that has been shared, and we'll get back to you. So thank you, everyone. Have a great day.